Hello, everyone. My name is David Danko. I am the Director of Bioinformatics and Software Engineering at Biotia. And today I'm going to be talking about how we can use molecular surveillance to better understand our cities and better plan public health responses using molecular data and microbial sequencing. So I always like to talk about a bit of context, a bit of historical context whenever I think about microbiology, because up until fairly recently, really just the last 20 or 30 years, most microbiology was done with a set of toolkits that would sort of surprise a modern day biologist training today. And that was largely just a microscope and a culture toolkit. And how this would go, many people are of course familiar with this workflow, is that you would take your sample, be it a single microbe, be it a microbiome, a collection of microbes, anything like that, you would plate it on a particular culture plate, and then you would perform some qualitative assays on that culture, you would perform some functional assays, you would basically look at this under a microscope and you would see what was there. And this kind of approach yielded a wealth of knowledge really about microbes. It was really incredible how much data this can pull out. It told us about taxonomy, it taught us about the basic functions of microbes, their structures, how they were organized into groups, some of their cooperative potential. But the downside of it, of course, is that it's a very labor-intensive approach to do anything with microbes, particularly once you're getting into microbiomes, largely of communities. It's very, very hard to get high throughput, large-scale data out of this. And of course, also there are all the problems that certain microbes are very difficult to culture, which somewhat biases the results. But nonetheless, we got a fantastic amount of information out of this. And this is sort of the fundamental root of microbiology, but this has maybe changed and adapted in the last 20, 30 years or so. And that's, of course, with the rise of sequencing, of why that adaptation, why that change has happened. There is, of course, the famous slide that virtually every bioinformatics presentation ever has had that shows that the cost of sequencing, the cost of DNA sequencing has decreased exponentially faster than Moore's law. And while that's certainly true, and while the massive decrease in the cost of sequencing has been an important part of the rise of sequencing for microbiology and the rise of metagenomics, it's also the case that the actual drop in Moore's law has also enabled the rise of sequencing as an assay, as a useful tool for microbiology. Because with the decreasing cost of computation, with the dramatically decreasing cost of sequencing, we can now sequence and process vastly more data than would ever been possible before, to the point that we can understand these communities with very, very low abundance organisms and very small stuff that requires very deep sequencing to get to, using just molecular data. And of course, as part of this also, the third factor that allows this rise of sequencing is the vast increase in the number of reference genomes. This itself is, of course, due to the rise of sequencing, but it kind of has a bootstrapping, a positive virtuous cycle effect, where the more sequencing we have, the more compute we have, the more genomes we have, the better results we can get, and the more useful sequencing is to microbiology. The workflow for this kind of sequencing is also fairly well known, I think, to most people here. You take your microbes, you take your microbiome, you pull DNA out of that microbiome, and then you align that DNA to known reference genomes. And once you align that DNA to known reference genomes, you can get some idea of what's in your sample, what the functional genes might be there, and what the taxonomy, phylogeny, makeup of your sample is. And this is, critically important. Massive insights have already been found with the microbiome, lots of interesting stuff. But the one insight I want to focus on right now, that I think is particularly important, maybe often underappreciated, is that there really isn't such a thing as a microbiome anymore that we're seeing molecularly. There's no point, there's no scale, there's no size where you can draw a really bright line between what is a microbe and what is a macrobe. There are life forms, or debatably life forms in the case of prions and viruses, that exist at virtually every single scale we can think of, down to just little bigger than a carbon atom, all the way up to the largest known organisms in blue whales, elephants, that sort of thing. And there's no point where you can draw a bright line and say, like, well, this is too small to be considered a microbe, and this is too big to be considered a microbe. There's really a continuum here. And when we use a molecular assay like sequencing and metagenomics to understand this, we get all of this at once. We can find DNA from virtually every kingdom, every order, every domain, every part of this size spectrum all at the same time. And we can use that to better understand our world and better understand the interactions between the really big things and the really small things, and how all that scales up. Analogously, also, we can kind of arrange this in sort of a food pyramid idea, this idea of using molecular techniques to understand sequencing data, because we have the things that we want to understand at the bottom of the data, and these are big things. These are things like ecologies, human health, organisms, medicine. These are things we want to understand, and we can begin to look at these from a perspective of how they're built out of biological molecules, biological genes. So that would be genes, viruses, plasmids, things that let us transfect it. Certainly things like antimicrobials are a great example here, because these are produced by genes and genes clusters in microbes typically. But then you can also break those gene clusters down into individual genes, individual operons, or let those individual operons become individual sequences that produce parts of proteins or full proteins, codons and the nucleotides, all the way down. So you get many different scales. And what we're really trying to do here is understand this large scale of life, this large diversity in the size of life that we're dealing with also a large diversity in the scale of molecules. So we're both scaling in the size of organisms we're dealing with, and we're scaling in the size of molecules that we're using to understand these organisms. And my work in particular has focused a lot on how we can understand one particular and one really big ecosystem, which is the ecosystem and the microbiome of cities and urban environments. So for about 5 years, I've been working with Metasub, and Metasub is the Metagenomics of Subways and Urban Biomes Consortium. The name is maybe a little bit antiquated from our early work, because we now do more than just metagenomics. We do culturing experiments, we do metatranscriptomics, we do even a little bit of mass spectrometry, that sort of thing. But basically, we're trying to understand microbiomes via the metagenome, and we're just trying to understand those in cities, and in particular, mass transit systems in cities. Not just subways anymore, we do buses, we do any kind of mass transit system that's out there. But it started with studying the subways, particularly in New York City. So Metasub is this big global consortium. We have about 60, 70 cities right now, and every year we have just an army, a small army of like 1,000 volunteers go out across all these cities, and they take samples from just anything in this urban environment they can find. The only real criteria these samplers are given is that we want them to look for things that appear to be abiotic. So we're looking at things that are like park benches, metal hand railings, concrete sidewalks, that sort of thing. We're typically asking these volunteers not to go sample, for example, like a puddle or, you know, the friendly neighborhood rat that they found or something. So we're looking at largely abiotic surfaces, and we ask these volunteers to sample things. And we have a collection of about 5,000 or so sequence samples right now. Most of these samples are collected from 2016, 2017, have been sequenced with paired 150 sequencing, and that's largely what I'm going to be talking about for the results today. Because one of the most striking things we find in this data is this rather abstract plot of species prevalence. So species prevalence is the fraction of samples where a given species is found. And what this plot is showing is the number of species that are found with a given prevalence. What's not terribly surprising about this plot is the big spike on the left side of the plot, what is labeled the peripheral microbiome, the peripheral urban microbiome on this plot, which consists of species that are found in less than 25% of samples. There are a lot of these species that are found in only a small fraction of samples, and this is sort of expected. This makes sense that there would be a lot of diversity in these microbiomes. What is surprising here, and what I think is sort of critically important and interesting to Metasub's work, is this bump on the right side, actually, what we term the core or the subcore urban microbiome. Because this says that there's a set of species, much more than you would expect by random chance and random assortment. There's a set of species that are found in virtually every single microbiome, urban microbiome sample we're looking at. In particular, there's a set of about 61 species that we find in 95% of our samples. And that includes samples from cities all over the world, sampled at different times across different years, and yet we still have this core set of species. And what is striking about that core set of species when you break them down into their groups is that they're, one, clearly phylogenetically organized. They come from 3 different phyla: Actinobacteria, Firmicutes, and Proteobacteria. Notably, you don't see the Bacteroidetes in this. There's no core microbiome that's the Bacteroidetes. Notably, also, you don't see any viruses or fungi that show up in this core urban microbiome. There are some in the subcore. Saccharomyces cerevisiae is, of course, in the cup core. That's pretty common. It just doesn't quite make the cutoff for a core microbe. But these are phylogenetically organized into 3 phyla. And what's possibly the most interesting feature here, in my opinion, is that the majority of these taxa, the majority of these core taxa that are found in 95% of samples are not human commensal taxa. Which is weird, because the only thing that you can really say about cities definitively, the only thing that's common across all cities everywhere in the world that there are no exceptions to, is that humans live in these cities. So the only thing that we really expect to be driving the similarity between cities is the fact that there are humans in these cities, and we would expect human commensals to maybe be found in every sample. But we find the majority of our core microbes are not typically considered human commensal species. Not to say they're never found in humans, just that they're not typically considered human commensal species, which gives us this kind of striking pattern that we can't quite explain why this is the case yet, but gives us this striking pattern of a non-human ecosystem that we believe is interacting and probably viable and performing interesting functions in the urban space. Now, the fact that this core microbiome exists is not to distract from the fact that there are indeed continental differences between different samples. So we have a fairly large continental divergence that you can see when we plot all of these samples on a UMAP, all of the taxonomic profiles for these samples on a UMAP. And that geographic difference largely separates East Asian samples, so samples primarily from Hong Kong, Japan, Korea, Taiwan, largely separates these samples from samples taken from Europe and North America. And we see a fairly big continental divide. We also see smaller clusters that correspond to sub-Saharan Africa, to Oceania, to the Middle East. And these clusters are mainly smaller and more distinct only because we don't have as many samples from this region. Our expectation is that we add samples from this region, these clusters will become larger and we'll start to see more clear continental divergence between all of these regions. And that speaks to maybe some interesting identifiable trends. And that's a concept that when we were researching this, when we did this research, we wanted to push a little bit farther. Because what we found is not just that there is a core urban microbiome that is conserved across a large subset of samples, but that there also appear to be certain signatures that encode certain properties of the cities from where a microbe is taken that are also conserved across the world. And what I mean by that specifically is that we can find a certain set of microbes that will reliably indicate whether a sample was taken from a city on the coast, from a city that has high population, from a city that has a warm climate or cold climate. And how that works basically is we trained a random forest model to predict a city's population, climate, whatever you would like really, whatever you can see on this plot here. We trained a random forest model to predict this from all of our data except for the data from one city. And then we asked the model to predict the labels for the held out city. We asked it to predict the climate, the population for samples from the held out city. And we were able to do far, far better than random when we did something like that. And what that tells us is there's some kind of globally conserved signature that indicates these features of a city, which we find to be really surprising, really interesting, and completely unexplainable at this point. Because when we looked into some of these are sort of abstract concepts. It's hard to say what microbes might be associated with population, for example. When we looked into this data, we thought that we would be able to explain some of these features really well. In particular, we thought that we would be able to explain what microbes were associated with coastal cities really, really easily. Because we thought they were going to be halophilic microbes. We thought they were going to be ocean-loving microbes. We thought that they were going to be stuff that's found in water normally. Makes sense that would be associated with the coast. It wasn't what we found though, not at all. What we found is that of the sort of top 20 most associated microbes with coastal cities, they were not, except for 2 of them, associated with water at all in any documented evidence that we could find. So at this point, our ability to explain why these signatures exist, why they contain the species they contain, is very, very limited. And this is one of the main ongoing areas of research here, is understanding why these signatures exist and how they have a mechanistic effect on their environment and how the environment mechanistically affects them. And with that kind of unknown sense, I want to then touch on the most important piece of unknown data here, which is the fact that most of the DNA in these samples can't even be identified. So the red bars here represent sequences that are aligned to anything in a genome at 80% average nucleotide identity across all of our samples. So what we see is that almost 50% of our data cannot be aligned to any sequence in a known reference database at 80% average nucleotide identity. And for the other bioinformaticians out there that's using BLAST, should be able to pick it up at that percent identity. And what that tells us is that there is a great deal of unknown DNA that comes from likely unknown or poorly characterized clades with new and novel genes that are out there in these urban microbiomes that are floating around us every single day, but that we know very, very little about. And some of that surprisingly even comes from fairly well-characterized regions like Europe and North America that we might expect to have less, often still have a very, very high proportion of unknown DNA. And now I want to sort of switch gears a little bit, because what I've been talking about, the research we've been doing on urban microbes for the past 5 years, was until pretty recently considered maybe a little bit abstract, possibly a little bit academic. The idea was that we were studying urban microbes, we wanted to understand how they impacted human health, but most of our research was centered on the idea that we might be tracking antimicrobial resistance, we might be tracking kind of subclinical effects, we might be able to explain a little bit about urban-specific diseases. But that a lot of these endpoints, a lot of this research would be kind of a ways down the line, that we wouldn't be able to right away understand what was happening in some kind of disease system, just because we felt that these major disease systems were not too common, not too present anymore. And then, of course, there was the COVID-19 pandemic. This really changed the way we were looking at all of these urban microbiomes, because it sort of radically altered this notion of microbial surveillance, particularly for the purposes of public health. And we were fortunate enough to actually take a bunch of samples during the pandemic that let us get a sense of what would happen to these urban microbiomes in this unprecedented social change. So what we saw there is this sort of surprising, rapid, and sustained drop in microbial diversity throughout the pandemic that we feel is probably largely driven by people not entering public spaces as much. So what you can see here is a plot of the microbial diversity, the richness, the number of species in the two busiest stations in New York City, the two busiest subway stations in New York City, over the 2 weeks leading up to the initial March shutdowns in 2020. And what we see in that time is a pretty steep drop-off in the species richness during those stations in that time. And if we look at this same trend over many different cities over a much larger time period, we see just the same drop. We see this big continuous drop over a fairly long time period that's happening all over the world. And this drop is corresponding with a drop in the fraction of human RNA that we're seeing in these samples, so that's just actual human shed that we see in these samples, and the number of human-associated microbes that are in these samples. So we see these big species richness drop-offs that we believe is probably driven by a decrease in humans, a decrease in humans interacting with public spaces. And this notion of being able to use molecular surveillance to understand something about a disease system is really what has then motivated a lot of the work that Biosha has done and the work that I'm now doing at Biosha. Because we took this idea of microbial surveillance, what you could do with it, and we wanted to apply it to the idea of detecting variants in SARS-CoV-2. Of course, variants have gotten a lot of attention lately with the rise of Delta, but the idea here being that if you can track the evolution of a pathogen, you can better react to it, make finer-grained public health decisions, possibly create pharmaceutical interventions in certain cases, and better understand what's happening. And so those efforts have been pretty prominent in a lot of the world now. Mostly we think about the US simply because that's where we're based. And these abilities to do sequencing surveillance has given public health officials the ability to understand what strains are present, what strains are emerging and could potentially become issues, and importantly, to identify variants that are of potential concern to them, variants that are in vaccine-targeted regions, like the spike protein variants that could have potentially drive increases in infectivity, drive increases in the ability of someone to catch the disease or have a severe reaction to the disease. So identifying these variants and being able to map them out geographically is I think, really going to be critical to public health departments, both for the rest of the SARS-CoV-2 pandemic, as well as into the future dealing with new emerging disease with the increase of globalization, increase of things like that. So how this works from Biotia is we've worked with Twist Bioscience to develop a hybrid capture to pull down lots and lots of SARS-CoV-2 DNA from a sample, or RNA, excuse me, from a sample, perform sequencing on it, and then what Biotia does as part of this is run it through our custom software COVID-Dx to identify as many different variants, different pieces of interest as possible that we can then provide to public health, hospitals, doctors, anyone that's making sort of health-based decisions about people, we can provide this information to. And this software, COVID-Dx, along with the Twist panel, receives an FDA Emergency Use Authorization for the detection of SARS-CoV-2, which of course we were happy to see that, as well as a lot of research use only endpoints that we had to assess lineage, to assess variants as well. Basically, the idea of all this is once we have our SARS-CoV-2 DNA that's been enriched RNA, SARS-CoV-2 RNA from a sample, we can sequence it, we can align it to the SARS-CoV-2 genome, we can pull out a bunch of different research endpoints from that. In particular, things like the variants that are found in the genome, all the little indels, SNPs, any kind of difference between the Wuhan reference genome we can find in our data, we can list out, we can identify easily for clinicians, for patients, for public health departments, what kind of genes those variants can occur in. And connect them with any clinical literature that we have available that shows if a particular variant is associated with any certain type of increase in virulence, increase in infectivity, increase in danger to people. And then we can also place samples within a lineage, showing where the other similar clades have been geographically, what kind of emerging clades are there. Of course, now we're mostly thinking about Delta and how we can identify Delta and the substrains of Delta in this. And this ability to place and identify variants has proved surprisingly important and prescient because it's actually helped us to identify novel variants already in New York City. So we took, as part of the validation of this sample, we took 71 nasopharyngeal swabs taken from people in New York City. And we were able to detect a set of conserved mutations in these samples that were not already known in GSAID. And of course, then the nice thing is that we can automatically put those samples back up in GSAID so that they're accessible and useful to the broader research community in general. And this has allowed us to better identify novel genetic variants and identify potential new strains. Well, very potentially new strains at this point. But to give this information really to public health officials, to clinicians, to any researcher that's interested in this kind of thing, and make it easy for them to assess where their samples are, what kind of new variants they need to look out for, and what sort of the trend in their area might be. And with that, I just want to reiterate what we've done is build a molecular assay that allows high-grain strainotyping and variant detection in SARS-CoV-2. This has been built by a really fantastic team here at Biotia, of which I'm lucky to now be a part of. And of course, it's been built in partnership with a lot of different people, and particularly Twist Bioscience, who did the wet lab hybrid capture component of this assay. And with that, I'm going to end the talk here. You can enter questions in the chat box here, and I will answer them as we get them. Thank you very much for listening to this talk.